Welcome to our channel. In this detailed YouTube video, we will explore the principles, techniques, and practical applications of column chromatography. Additionally, we will delve into the crucial aspect of solvent selection, which plays a significant role in the success of the chromatographic process. Introduction to column chromatography. Column chromatography is a powerful separation technique used in the field of chemistry and biochemistry to separate and purify compounds from complex mixtures. It is based on the principles of differential adsorption and partitioning of components between a stationary phase and a mobile phase. How does it work? Column chromatography involves a column packed with a stationary phase material, such as silica gel or alumina, and a mobile phase that flows through the column. When the sample is loaded onto the column, each component interacts differently with the stationary and mobile phases, leading to their separation as they travel through the column. Advantages and applications. Column chromatography offers several advantages, including its versatility, simplicity, and ability to handle both small and large sample quantities. It finds applications in various fields, such as pharmaceuticals, natural product isolation, environmental analysis, and food science. Types of column chromatography. Normal phase chromatography. In this type, the stationary phase is polar, and the mobile phase is non-polar. Polar compounds have stronger interactions with the stationary phase, leading to their separation from non-polar compounds. Reverse phase chromatography. Here, the stationary phase is non-polar, while the mobile phase is polar. Non-polar compounds interact more with the stationary phase and elute first, separating them from polar compounds. Ion exchange chromatography. This method utilizes charged stationary phases to separate compounds based on their ionic interactions with the stationary phase. Size exclusion chromatography, also known as gel filtration chromatography, it separates compounds based on their size. Larger molecules elute first, while smaller ones are retained longer. Column packing. Choosing the right stationary phase, such as silica gel or alumina, is essential for successful column chromatography. The two common packing methods are the slurry packing method, where the stationary phase is mixed with a solvent to form a slurry, and the dry packing method, where the stationary phase is added directly to the column. Choosing the right solvent. Solvent selection significantly impacts the success of column chromatography. The solvent's polarity, solubility, and compatibility with the stationary phase are crucial considerations when choosing the mobile phase. Common solvents used include various organic solvents and mixtures. Mobile phase composition. The mobile phase composition can be either isocratic, constant composition, or involve gradient elution, changing composition over time. The mobile phase composition affects the separation efficiency and resolution of the chromatographic process. Sample loading and elution. Preparing the sample for loading involves dissolving it in a suitable solvent and carefully loading it onto the column. The elution process involves the flow of the mobile phase through the column, resulting in the separation of components based on their interactions with the stationary phase. Troubleshooting and tips. During column chromatography, common issues may arise, such as tailing peaks or poor resolution. Knowing how to troubleshoot and optimize chromatographic conditions is essential for obtaining reliable and reproducible results. Applications and examples. Column chromatography finds applications in various fields, such as isolating natural products from plants, purifying pharmaceutical compounds, and analyzing complex mixtures in environmental samples. Practical examples and case studies showcase the versatility and significance of this technique. Safety considerations. Handling solvents and chemicals in the laboratory requires proper safety measures to ensure a safe working environment. Adhering to safety protocols and wearing appropriate protective equipment is essential during column chromatography. Let the applications and examples we've explored inspire you in your own research pursuits. Remember that each chromatographic separation is an opportunity for discovery and innovation. Thank you for joining us on this incredible journey. Remember to like, subscribe, and share this video with fellow science enthusiasts, so together, we can spread the wonder of column chromatography far and wide. Until we meet again on our next scientific odyssey, keep exploring, keep learning, and keep pushing the boundaries of knowledge. Happy chromatographing!